welcome to a quick video on binary code and DNA. Let's see if it will load. There we go. So if you haven't seen, binary code is the code in which computers and technology operates, and it's a 0 and 1 function. It is black and white, yes or no, up or down if you want to think about that. Now, the reason why we're talking about binary code for homework tonight is you're going to watch this video. I'm not going to play it now because it might screw up with the microphone. And it talks about binary code, and I want you to watch it and do your best to answer 4 questions on the back or the front of your note sheet, which has those tables there. We'll talk about them in a minute. This is DNA. DNA is the genetic code of all living material. Viruses have it, bacteria have it, fungi have it, whales have it, we have it, parrots have it, plants have it. Anything that has lived on planet Earth and possibly outside of planet Earth has a genetic code that is inheritable and codes for traits and proteins when it is read and expressed. Just like zeros and ones on your phone might lead you to see a video or a picture or a text, different information from different sources, but all interpreted the same way. So there are 4 questions I want you to think about as you watch this video about binary code. You probably have very little background in binary code, yet it is the thing that makes our world run. The first thing I want you to think about is how does the code achieve a stable 0 or 1 output? 2, how does the rule influence how a code is interpreted? 3, how is context important for looking at a code sequence? And 4, how is the code or sequence able to be compressed? So I'll put this link to the video in the Schoology assignment so you can see it. Let's talk about these answers for DNA to give you a foot into the door. Oop, that is not going to show what I want it to show, so I'm just going to quickly move out and put this over to the left. Sorry about that. Good thing. What you need to know about DNA in terms of this question is that DNA is coded as either one of 2 molecules, which we call a purine or a pyrimidine. So again, it's a binary code. Both have very different structures, so when a purine is present or a pyrimidine is present, enzymes that read DNA to make proteins code it as either a 0 or a 1. Now, when we look at the images on the back of your note sheet, you'll note that for purines, we have 2 different options. Think of it as 0A or 0B. We can have an adenine or a guanine that make up our purine option. If we're thinking about our 1 option, 0 or 1, black or white, we have a molecule known as pyrimidine. They tend to have a smaller molecular structure. We call these structures nitrogenous bases. And we have 3 options depending on the molecule that you're in. Cytosine is found in DNA and RNA. We'll talk about the differences between those 2 later on in this unit. We have thymine and we have uracil. Really, you can think about this as 1A or 1B, 0A or 0B. So we have a binary code for DNA as well. Second question, how do the rules influence how a code is interpreted? DNA that codes for genes, traits, eye color, hair color, dimples, freckles, left-handed, right-handedness, your skin tone, your proclivity towards having obesity or diabetes, are genes that are regulated, and they tend to have things that regulate their expression, whether we promote them or inhibit them. This influences how genes are read in different cells, and those cells might express those genes at different rates. When we look at the structure of chromosomes and we look at big coding sections of DNA that make genes, we tend to have things like operators and promoters, which are segments of DNA that don't make a protein, but rather influence whether the gene is expressed or not. So in this example here, in a situation in which there is no lactose present in a cell, lactose being a milk sugar, we repress the gene that would make lactase the enzyme. If we don't have firewood, we don't need to burn the fire. Gene expression is dependent upon the environment. But in an alternative cell that has maybe digested a bunch of milk from a milkshake or had their first creamy, We use different enzymes to make the gene be expressed, to make the lactase so that we don't have a stomach ache. Thinking back to this question, we have rules that influence whether the code is interpreted, expressed, or even prohibited. So our third question, how is context important for looking at a code sequence? When you watch this video about binary code, zeros and ones can code for text messages or musical sounds or a visualization of a picture. How does that apply to cells? Well, the same genes code for molecules that might regulate other genes. So we might have some genetic information that make things turn on other genes. At the same time, we might have a similar collection of DNA that makes genes for proteins. Maybe they make enzymes or transport proteins, or maybe they make structural proteins. A lot of DNA is just random. It's there to make the structure of our chromosomes work. They're not really doing anything for us. So the context of DNA really is dependent upon, is this DNA expressed or not? Is it structural or is it genetically coding? What chromosome do we find on it? A bunch of zeros and ones doesn't mean a lot until we think about what its job is and what it does for our cells. And that's even true when we think about the fact that every cell in your body has the same chromosome. Here's chromosome 7, and it has a bunch of different genes that may or may not be expressed in different cells. So for example, EPO, the gene that makes erythropoietin, it makes red blood cells, might be expressed in our red blood cells, our kidney cells, but we're probably not going to be expressing this gene in our irises or our neurons in our brain. At the same time, epidermal growth factor, epidermal being your outer skin, might be a gene that we express in your skin, but definitely not in your muscle cells deep inside of your body. Different genes might be expressed differently in different cells based on their environment. And then finally, for this last question, how is the code able to be compressed? We want to be able to compress our code for easy mitosis, easy meiosis, and also regulate gene expression. The regulation of gene expression is whether or not we make that gene available to make a protein, or do we turn it off. DNA can be methylated or acetylated to make DNA accessible, or basically heard. This allows us to make proteins. When we methylate molecules, we are minimizing the space between DNA. We're generally closing it off. This is what we called heterochromatin during our mitosis unit. This is the DNA that's not available to make genes or make proteins. And this is the DNA that we tend to use for prohibiting gene expression or for mitosis and meiosis. Alternatively, if we acetylate or give access to, methylate, minimize, acetylate, access, we have DNA open, and then we can express it. We can make traits, we can make genes, we can do stuff with it. Acetylate, action, accessible. Maybe those that alliteration helps you. Methylate, minimize. Can't think of a third M word right now, I apologize. That is how we can condense, compress, and regulate our DNA. So we will talk more about each of these ideas over the next couple weeks, but this is a starter taste as to how we can take our genetic code and do a lot with it to make a code that is very simple, but very elegant, and very easy to be environmentally responsive. I hope you come in with questions tomorrow. Enjoy the rest of your sunny day today.